Hello guys, welcome to Mr. Ghost Directors. This director is very important to a student who wants to achieve his dream. Today's director majorly going to focus on something called the hormones. You get? In the very lectures, we looked at something called the endocrine system, and we saw how we have several glands in the body, and these glands here they secrete a chemical substance which is called the hormone. You get? So we're going to break down this director here, and then we're going to be looking it down like this here. The first component is going to be looking at now. The first part is going to see what we call now the definition of the hormone. Definition of the hormone. Hey guys, what is a hormone members here? A hormone is a chemical substance that is secreted by a specific cells or specific group of cells which are from the glands, and then it is released into the bloodstream. So that means that this hormone here is going to be secreted by what we call structures called the glands, and this hormone here is going to be released in what we call the bloodstream. This is the bloodstream. And then the bloodstream here, this hormone is going to be transported into what we call the target organ. The target organ. And most of these are the cells members. These are the cells that make up the organ. These are the cells of the organ members. And now these cells here, they bring about a change in the structure or in the functioning of the organism. You get? So a hormone is a chemical substance that is secreted in what we call the gland and to the bloodstream, and then they are transported into the target organ where they cause several cellular functionings. Now, here we say the cell in the organ they're going to cause what we call cellular functionings. Cellular functionings. That is something very interesting, members. And then the second part, we just are going to look at something which is going to be very exciting here. I'm going to press it here. This exciting part we're going to look at number 2 here. This is going to be now, we're going to break down these hormones here. We can say classification of the hormones. Classification of the hormones. Remember here, we classify these hormones here based on 2 major components now. We based on what we call the binding site. One is going to be the binding site for these hormones here. The binding site. So it's going to be what's called the binding site. That's going to be the first part of this hormone is here. Going to be the binding site. And the second is I'll see what we call the structural composition. You can say the structural composition of these hormones here. Structural composition. Remember, listen here. We have 2 types of what we call the hormones here. We have 2 types of what we call the hormones. The first type of the hormone is going to be called the peptide hormone. We have what to call the first part is going to be called the peptide hormone. It's called the peptide hormone. The peptide hormone. Then the second part is called the steroid hormone. Steroid hormone. How do you classify them here? We say you are classifying them based on what you call the binding site and also what you call the structural composition. Remember, so you look at these peptide hormones here, if I can bring them here, they have what you call interesting features. The peptide hormones have what you call interesting features here. When you look at them, number 1 is that they are highly soluble in water. So we can say they are soluble in water. Soluble in water. That means you can say that they are also hydrophilic since they are soluble in water. That means they are also what? Hydrophilic. So they're hydrophilic. Then they are soluble in water. Say they're hydrophilic. And also when you look at their structural composition, remember these proteins here, they have a structure which is a little bit complex. So we say it is a complex structure. We can say complex insoluble structure. And then I will look at now what's going to happen here, the binding site. Why the binding site for this peptide hormone here? The binding site for this peptide hormone here, they are major where. Let's look at it here. The binding site for this hormone here, they are actually the binding sites are supposed to be they're on the surface of the membrane. You can say surface of the membrane. So these peptide hormones here, they do not pass through the cell membrane. Since we say they do not, because the cell membrane, look at the structure of the cell membrane here. This structure here is majorly a phospholipid bilayer having what we call a phospholipids, and these phospholipids here do not allow any peptide molecule to pass through them. So if they want to go inside, they must bind onto the receptors on the surface, which are going to send the signals, and then the signals are taken inside the cell. So we say their binding site is on the what? On the surface, not inside. You get? Whereas when you look at something called now the steroid hormones here, these steroid hormones here, one is that they are done what? They are highly soluble because they are lipids in nature, because they're majorly made of what we call cholesterol. So we say they are highly soluble in the phospholipid bilayer. That means soluble in the phospholipid. I can say the cell membrane. You can say soluble in the cell membrane. Soluble in the cell membrane. And then you can say they are, though they're hydrophobic, they also structurally, we can say they are what? They're hydrophobic. Reason being they do not dissolve in water, but they are soluble in lipids only. You get? So that is what you call now the peptide, what you call the steroid hormones here. Peptide hormones soluble in water, but not the lipids, and also they have what you call structural composition that is highly complex, that it has large particles, large molecules that cannot pass through what we call the phospholipid barrier. And secondly, its binding sites on the surface of the cell membrane. So we can look at some of the examples of the hormones here that are under what we call the peptide hormone here. We have what we call the peptide hormones. We have what we call some of them what we call, we have what we call the oxytocin hormone. We have what we call luteinizing hormone. We have the epinephrine. We have glucagon, and very many others. Whereas on the side of what we call the steroid hormones here, we have what we call the steroid hormone. Steroid hormone, guys. Our dressing hormones, we have called aldosterone is one of them. Most of the sex hormones, most of them, we have aldosterone is one of them. And also made what we call cholesterol. Then also have what we call the testosterone hormone, and very many others. So we have what we call, these are called the steroid hormone, what we call what? The peptide hormones here. But now the second part that you're going to look at, members, we have seen the definition of the hormone. We have seen what to call the classification of the hormone here. This was B, this was A here. Then we need to see that we say that these hormones here, they normally pass and they normally go and bind on the given receptors. That means we don't look at what we call the binding sites called the receptors. We have what to call the binding sites, which are called the binding sites. Which is going to be C, called the receptors. Remember these binding sites here, we're going to see most of them are going to be called now. We have what to call, these are called receptors. I mean, these receptors members here, they have majorly three components. They have in the part that is on the surface, if this is the receptor, it is having this surface here. They also having what we call this one down here. This is the cell membrane that you are seeing here, and then we have this the receptor. So the part that is outside the receptor here is what we call extracellular domain. This is called extracellular domain. Extracellular domain. Now the one we are seeing here that is within the cell membrane is what we call the transmembrane, or the hydrophobic domain, transmembrane receptor domain. Then we have the one that is inside here, this one that is within the cytoplasm. This one what we call intracellular domain. That's very interesting, members here. Of course, you have very many receptors, members. You have what to call some of the receptors to the surface of the membrane. You can say some of the receptors, examples of the receptors, examples of the cell surface receptors. When we have the interesting receptor here that we have here, one of them is going to be called the G protein coupled receptor, something called the G protein coupled receptors, members. Here we have to look at it. We have what called the G protein coupled receptors here. This is the G protein coupled receptors here. We have what called the calcium. We have what called the calcium. Okay, sorry. You can say the iron channels. Can you say ion channel receptors? Ion channel receptors. We have what called this one is for the protein coupled receptor. This one here, it is major important in what you call it is major important in activating what you call the enzyme adenine cyclase and then bringing about phosphorylation. You get? So it is important what you call metabolism, the glucose or the cellular metabolism. So you have what you call ion channels. This one is normal open. They have the gates, the normal open, and then they allow the intake of the materials here. Then secondly, we have what you call the third. We have what you call the cyclic MP. Okay, sorry. We have what you call the enzyme linked receptors. Receptors here. These ones are very funny. They have what you call on their intracellular domain here, like here. They have what you call they are connected to an enzyme or they are enzyme in nature. These are called the enzyme linked receptors here, members here. So we have seen a brief about the first what called hormone from the glands to the bloodstream and then to the target organ, causing what called cellular functions. And then we have seen what called classification of the hormones here, based on the binding site and also the structural composition. That we have what called the peptide hormone, we have what called the steroid hormone. You get? So, for example, we have what we call the oxytocin, luteinizing hormone, epinephrine, glucagon, and very glucagon, insulin, and very many others. Then the steroid hormone, we have what we call testosterone, and very many others. They are binding on the receptors. Which receptors here? Such a receptor has three components. It has the extracellular domain, transmembrane domain, and also what we call intracellular domain here. Then you see that example what we call the cell surface receptors here. We have what we call the G protein coupled receptors. We have what we call the ion channel receptors, also what we call the enzyme linked receptors here. Let's move on, members here. In the next part now we're going to say something very interesting here. We need to see that, or we know that most of the chemical substances, remember here what normally binds here is called the regard. This can be called the regard. What binds on the receptors is called what? The regard. This can be hormone. This is called the regard. It can be hormone, can be any chemical substance, can be a neurotransmitter. You get? Now let's listen here. There's something we normally call the first and the second messenger. If this is my cell here, this is my cell here like this. The cell is having what you call a given receptor here, which is having what you call extracellular domain, and then intracellular domain here. This is my receptor. This is my what? My receptor. This is the cell membrane, of course, that you are seeing here. This is the cell membrane. Now, when the regard binds here, this is the regard binds here. This could be the hormone. For example, it could be what you call the epinephrine. When it binds, what's going to happen here is that this epinephrine, this hormone here, the receptor is going to detect the signals from the hormone. Now the detection of this hormone here by the receptor here, it means that the receptor has seen the first signals. In other words, the first message has been sent. So this has got what called the first messenger. The binding of the extracellular, of the hormone onto the extracellular signal is going to cause what to call the second. This is supposed to be called the first messenger. And what is the first message has been gotten. This is called the what? The first messenger. Now, after binding here, the signals are going to be sent to what we call the transmembrane. This is called transmembrane receptor, and then I'm going to go to this what we call the intracellular domain. Now, this cellular domain here, remember we have several proteins that are going to be here. It's attaching on the several proteins here. Now, when these signals reach on the domain here, on this protein here, this is a given protein. This protein is going to become activated, or it is going to be stimulated. You can say stimulated. So when the protein gets stimulated, what's going to happen here is that it is going to begin sending these signals here to several proteins inside, to several enzymes inside, and some could be in the cellular membrane, some could be inside the cytoplasm. So the signals are going to be amplified, whereby some can even go into what we call what? The nucleus, whereby they can cause several processes like transcription, protein synthesis can take place. So the signals go on amplifying. Now, from this protein here, which has been excited, and then beginning to amplify the signals, this is called the second messenger system. It's called the second messenger system, members here. It is called the second messenger system. So the second messenger here is whereby the signaling mechanisms that are coming from what we call the extracellular environment, they amplified. In other words, the first messenger, in other words, the first signals whereby we amplified. Here in the second messenger, we amplify the first messenger signals. You get, these signaling molecules are amplified so as they can bring about a given response or cellular response. You get. Now we have very many messengers here that are inside here. Most of these are called the messengers. These are called what? Messengers. These are called what? Messengers. Now these are called what? Messengers. Well, these are what? Messengers. We have very many messengers members. One of them what to call the car. That's what to call some of the messengers that we have. One of them is what to call the calcium. That's supposed to be the calcium ions. It's one of the messengers that we have there. So it's majorly for the muscle contraction or a neurotransmitter release of the neurotransmitter. Then also have what to call another one. This one is for muscle contraction. Muscle contraction. And also neurotransmitter release. Neurotransmitter release here. Then also have another one which is called now the diacyl glycerol, also giving us that. You get, this one is majorly important one to call. It is covalently phosphorylating. In other words, activating the protein, the phosphokinase A or the protein kinase A, and then so as it can easily phosphorylate several proteins so as they can be able to manufacture enzymes, be able to open the channels of the cell membrane and so on. They also have what to call something called IP3. This is inositol triphosphate. That's what called inositol. Inositol, that is 1,4,5 triphosphate. Of course, we have others like the cyclic AMP. Also, what called the cyclic GMP, and then We have very many of them and very many others that we have in the body. So these are what called the secondary messengers. They are very important inside the cell. They are good at trying to to amplify the signal so as they can bring about a given response in an organism. Now, I want us to see the major focus of today. We want to see how is the signal moved majorly across the cell. For example, if the hormone, maybe if I have less glucose, if I have less glucose in the blood, what happens? Something very interesting here. We shall see the hormone is going to bind onto the cell membrane, if it is peptide hormone, and then it's going to go inside and cause several changes. Let's look at it here, something very interesting. Now, the second part, we're going to begin looking at now the action of what called the peptide hormones. We shall begin by looking at what? Peptide hormones. So here we're going to look at something called the peptide hormones here. In other words, we are going to begin breaking down something called the peptide hormones. It's going to be our focus here. The peptide hormones. First. Listen, members here. Now what we're going to focus in here, we are going to look at the mechanism of action. In other words, the mechanism of action of these peptide hormones here. The mechanisms of actions. Can also be called the pathways. The hormonal pathways. Can you say the hormonal pathways? So here we're just going to measure, look at interesting things that you're going to measure involve here. One, we shall involve something that is called now, we shall have what called the G protein coupled receptors. It's going to be one of them. We shall have what called the G protein. It's going to be one of the areas we're going to emphasize there, the G proteins. We shall have what to call something called the GDP and GTP. We shall have something like an enzyme called adenine cyclase enzyme. We shall have what to call a given protein which is going to become what to call the protein. It's going to be called a protein kinase A. We shall see something like also the protein kinase C is also going to be there. You get? We shall look at very many messengers that are going to be involved here. Messengers like the IP3. We shall look at some IP3. We shall see this IP3. Then we shall see something like the calcium ions. We shall see something like the messengers that are going to participate there. Then we shall look at also some lipids within the cell membrane. For example, the PIP2. Yes. So we see also the DAD. This one is diacetyl. This one is a messenger taking place there. And then we shall see the products like cell metabolism. We shall see also these are the results of the outcome. We shall see what we call the protein synthesis. We shall see like the cell division. We shall see the nerve. We shall see what we call the muscle contraction. And very many others. So now that is what we call now. So in the next part now we're going to look at now how the peptide hormones are going to moving across the cell. So now we are going to look at how this peptide hormone is going to be transmitted. We'll give you an example here. We have this is the G protein coupled receptor that we are seeing here. This is called the G protein coupled receptor that we are seeing here. You get? It is having what we call the extracellular domain here, and also has what we call what? Intracellular domain. Let me say the intracellular domain is here. Intracellular domain. This one is intracellular what? Domain here. And this intracellular domain here is bound onto what we call the G proteins. The G proteins. Check out the G proteins that we're having there. So what happens is that you are having the hormone, for example, epinephrine is going to bind onto what we call the extracellular receptors here, and then the signals are going to be sent. After the signals reaching here, they are going to be detected by something very interesting here. They are going to be detected by something very interesting here, by this G protein here. What's the name of this G protein? The G protein, remember you have what to call the GP protein and also what to call the GQ protein. You have two areas. So you have what to call the GP protein. Then you have what to call what? The GQ protein. So this one what to call the GP protein. So the GP protein members has components here. It is having like three subunits. It is having the first unit here, and the second and the third unit here. This one is called alpha. The mega. The gamma. This is called the beta. This is called what? Alpha. Sorry, it's called alpha. This is called what? The gamma. Now, the subunits that we have here, the subunits, sorry, bodies. Now, these subunits here, they're the most stimulating part of the G protein. Now, these subunits here, what they have is that they have certain kind of the components, energy stores. They have what to call the G or not here. They are bound to something called the GDP. You get? So what happens is this, members. When a hormone binds onto the receptors here, the hormone is going to send signals. Whenever signals reach on this G, this is called the G protein. This is called the G protein. What's going to happen here? We have what to call GDP here. So this G protein here is inactive. We say it's non-stimulatory. It is inactive. Or can say it is inactivated. Whenever there is a GDP here, it's inactivated. So how to we activate it? The moment we disperse the GDP here with GTP, then it's going to be what? Active. What's going to happen here? Now, remember in the cytoplasm here, we have what to call a lot of GTP in the cytoplasm here. They are in a high concentration here. You get? We also have another given enzyme which is called the GEF. We have what to call a factor called the GEF. Guanoside. This is what called the guanoside. Let's go the factor, nucleotide, exchange factor is what we call the guanoside exchange. So guanoside nucleotide exchange factor. Remember this factor, members, is always located on the cell membrane. You get? This factor, what called what? Is on the cell membrane here. This is the factor GFE. So whenever any stimulus reaches the cell membrane here, and then it hits on this G protein here, in the same vein, this GEF is also what? Is also stimulated. So it is going to move towards the G protein, and then it's going to cause this tightly bound GDP to be scrapped off, to be removed from what called the G protein. So this is going to be removed, and then the GTP is going to substitute and occupy where this GDP was, and hence we shall have something in this format here. We shall have a structure like this. Shall have a structure like this. With a GTP located here, GTP located here. Then we shall have a blockade structure of what to call this is a G protein. So I have what to call the alpha structure like this, separated from what to call this component of the beta and the gamma. So the structure has been distorted. You get to see it is now activated. This one is now what? Activated form of the G protein. So this activated form of the G protein, what is it going to do, members here? It is going to move onto where? It's going to move onto the cell membrane, and it's going to bind is going to bind onto, there is a given domain here, there's a domain here, so it's going to bind onto this domain here, so it is going to attach on this side here, it's going to attach here, so it shall have something like this attaching, but with a connected what? This is the GTP. It's activated. Of course, this is like this. Now, what's going to happen? Remember that when it reaches here, when it binds here, it is going to, remember this one is activated. It's going to activate this enzyme here. What's the name of this enzyme here? This enzyme is called adenyl cyclase enzyme. It's called adenyl or adenyl cyclase enzyme. So this enzyme is going to be activated. So after being activated, what it's going to do here, remember the cytoplasm here, we have what called a lot of ATP. You get what it's going to do? This enzyme here is going to dephosphorylate this ATP here, format called the cyclic AMP. Cyclic AMP, that is cyclic AMP. Now this cyclic AMP here, what is it going to do here? It is going to further activate a given enzyme. What's the name of this enzyme here? It's called the protein kinase A. We have what called the protein kinase enzyme here, which is located here. It's an enzyme here, located here. So it is going to activate this enzyme here. What's the name of this enzyme here? This is called the protein kinase enzyme. Now this protein kinase here, whenever it is, and what is it going to do here? Remember, we are sending signals. This one, we say this one is a messenger. It's a messenger. What else is a messenger here? Of course, this G protein is also what? A messenger. The G protein is also what? A messenger. So, immediately, this enzyme here converts the ATP into cyclic AMP here. It immediately produces a given enzyme. There is a given enzyme called the GTPase enzyme. The GTPase enzyme is going to be separated by this one here. It's called the GTPase enzyme, or the GTPase enzyme. So this enzyme here is going to majorly remove this phosphate from it. It's going to dephosphorylate it, and then we remain what to call the GTP is going to be removed. In other words, it is going to be converted into what to call what? GDP. So it is now inactivated. So it is switched off again. So the moment this one is converted into GDP, that means we say it has been what? Switched off. At first it was on because it was activated. But the moment the enzyme converts or dephosphorates this GTP into what we call the GDP here, it goes back to the normal state. So we say it has been inactivated or switched off. But also, maybe something to note down is that instead of using the GEF, the guanosine nucleotide exchange factor, it can also be done physically. We can add a phosphate on what we call phosphorylation or direct phosphorylation of the GDP. Instead of this one here, you can have what we call direct what? Direct phosphorylation. Phosphorylation. Whereby a phosphate is going to be on what we call what? The GDP, to form what we call GTP. Without the GDP leaving the what? The G protein. Using some enzyme, the nucleoside. You get kinase enzyme, which is going to do that role there. Let's get back here. We are still looking at this the protein kinase A. This kinase on here is going to do several processes here. Remember, it is just going to do the process called phosphorylation. Phosphorylation is the one that is going to do majorly. phosphorylation is what it's going to do. Phosphorylating what now? Most of the metabolic reactions. Now, one is that we have the nucleus here. Of course, we have, remember, we have what we call intracellular binding sites. In other words, the hormones can bind inside the what? The cell, or outside the what? The cell. So what's going to happen is that this protein kinase A here is going to, we have what's called the DNA here. This is the DNA located here. What's going to happen is that this protein kinase A is going to look at transcription factors, one of the transcription factors which are here. Transcription factors. It is going to come and activate them. It's going to add a phosphate, you can say phosphorylate them. Phosphorylate the transcription what? Transcription factors. These factors are going to move inside what? Inside the nucleus. This is the nucleus you're having here. So this nucleus here, after moving inside here, they are going to go to the DNA, bind to the DNA on the section of the DNA here. After what's going to happen after binding there, this DNA is going to, they are going to send signals. They are going to activate what we call the RNA polymerase enzyme. And they're going to cause what we call now the conversion of the DNA into what we call the messenger RNA. So we have what we call, this is going to be converted to call the messenger RNA, which is going to leave the nucleus and then moves on to the what? Cell membrane. And then undergoes what we call translation, whereby it's going to command to call some ribosomes here. This is going to be mRNA what we call with some ribosomes here. Then you form what we call now a given protein. You get? So the proteins can be what we call one, can be enzymes. You get? One, this, for example, enzymes. Most of these enzymes here, they can be important to call the glycolysis or glycolytic process can be important to call glycolytic or glycolysis to give out to call what? Energy. For example, some of the enzymes here can be used, for example, that is giving energy. And also this phosphate here can go and phosphorylate what we call some enzymes in the muscles. We have, this is the muscle, this is the muscle here, this is the muscle. So what they do, they are going to phosphorylate this muscle here. And then there's an enzyme called phosphorylase. Phosphorylase. This is a phosphorylase B. Is going to be activated. This is the muscle and also the what? The liver. So these the enzymes which are here are going to be activated. After activating them, they are going to be, it's also here and also here. It's going to cause the conversion of the glycogen in these structures here into what? Glucose. So this protein kinase can also go on the cell membrane here, can also go on the what? On the cell membrane and try to activate some kind of the what? Of the protein channels here. It's going to activate them, protein channels here. The gates that are here, the ion channels here, is going to add a phosphate. So when a phosphate is added on this these channels here, what are they going to do? They are going to open and then the ions are going to enter, the molecules are going to enter. For example, if this calcium can enter, sodium can enter, and some, if possible, can what? Can leave. The ions can leave and so on. So it is phosphorylating several reactions. For example, glycolysis, and also maybe formation of the proteins, transcription, and so on. You get it? Now, that one is finished now. Let's look at now, what if the hormone, let me say the hormone, is now as bind on another, this one was the G protein receptor, the GP protein receptor. Now what if it binds on what we call the G2 receptor? Something very interesting, members here. Something very interesting. Now, if the hormone has bound onto the G2 protein receptor, what's going to happen? So, now we're going to see now, look at now, we have seen the G protein coupled receptor, and then we see the G protein. Now, what if this is the same? It's the same as the G protein coupled receptor. But here, the intracellular domain is being bound, is attaching intracellular domain. Is attached on the GQ protein coupled receptor. That one is, I mean the GQ protein. GQ protein. So this GQ protein, what's going to happen is that it is going to be just like the same process, like the same here. It is going to be the GD is going to be what? The GDP is going to be removed. You get? Then we form what to call the what? We form what to call something called what? Something called, this is going to be the GTP. So this one what to call the G2 protein. Now this one what to call, so there's what one, this one, this is inactive form of it. This one is the activated or the stimulated. What happens is that this stimulated one is going to just go on the membrane. It's going to go on the membrane here. It's going to go on the membrane. Let me say here we forgot something like, let me say this is the hormone. This hormone here could be maybe any hormone. It can be the hormone for contraction of the muscles. Which hormone is that? So you can put oxytocin hormone. Can you put the oxytocin hormone has binding to this extracellular receptor here. After binding here, signals are sent. After sending the signals here, of course, there are very many domains. Signals are sent here, and then the intracellular domain is going to activate the G2 protein and form out to call activated the GTP protein. Now, what's happening that we have a given type of an enzyme that is on the membrane here. The membrane here, we have a given enzyme. What's the name of that enzyme, members? There's an enzyme here. So this is the enzyme that is lying on what we call this structure here. This enzyme is called the phospholipase enzyme. It's called the phospholipase C enzyme. Phospholipase, that is lipase C enzyme, PLC. Now what happens is that this is going to bind onto this enzyme here, because the enzyme has some binding sites on it. This is going to bind onto this. What's going to happen? This enzyme is going to be activated. What happens when the enzyme is activated here? This enzyme is going to cleavage, or it's going to break. We have some kind of the lipid molecule which is here. There is a lipid molecule which is here. What's the name of that lipid molecule, members? There is a lipid molecule that is here. Let me write it using this. There is a lipid molecule which is here. Lipid molecule here. So this enzyme is going to begin blocking. It's going to begin blocking this lipid molecule here. What's the name of this lipid molecule here? It's called the PIP2 phosphatidylinositol supposed to be 4,5 triphosphate. So this one is going to be blocking down. It's going to be blocking down into two molecules, into two messenger smaller molecules here. One is going to be called the what? It's going to be called the DLD, and then the second one is going to be called the IP3. The IP3. So this IP3 members here, it just moves away straight to what we call the endoplasmic reticulum. That's where it is having its binding sites. If I'm having my structure here of the endoplasmic reticulum, something very small here. I have my endoplasmic reticulum here. Let me say this here, this side here. Endoplasmic reticulum is here. Now, this is what we call endoplasmic reticulum. It can also be called the sarcoplasmic. It is in the muscles. Sarcoplasmic reticulum, it is in the mat. If it is in the muscles. what happens that on the membrane here, there are certain kind of the channels, there are certain kind of the binding sites for this hormone here. So called the binding sites for this hormone here. Then the same hormone has some kind of the opening gets here. So what happens is that this hormone here is going to bind onto this. After binding, it's going to activate this receptor here, and then the receptor is going to open. So it is going to open these channels here. Are going to be made open. Channels are going to be made open. And then what's going to happen here, we shall see, remember there is a lot of calcium ions here. You have a lot of calcium ions here, a lot of calcium ions. So the calcium ions are going to leave. So what's happening when the calcium ions leave, members? It's just going to go. There's something called the cadmium. There's a given kind of protein called the cadmium here, which is going to move, and then they are going to be detected by the cadmium. The cadmium is going to detect them. After detecting them, it's going to do what? It's going to be activated, and then it is going to activate the kinase enzymes. Kinases, these enzymes here. These enzymes here are going to do what? Are going to do what we call various phosphorylations. The various what? Phosphorylations of proteins. Whereby they are going to phosphorylate, just like the same way. They are going to do what? They are going to phosphorylate. They are going to add the phosphate to what to call the enzymes. That means they are going to activate what? Activate enzymes. Because there are some enzymes that are not activated, the precursor enzymes. They are going to phosphorylate the channels. That means the cell membrane channels or the protein channels. They are going to do the same thing. What are they going to do? They're going to do something very interesting here. They are going to also activate certain kind of the metabolic enzymes, like the phosphorylase B into phosphorylase A. That means we say activation of metabolic enzymes. They can also try to activate some enzymes that are possible for the breakdown of what to call the lipids. That means lipolysis. Lipo is here. Lipolysis. The breakdown of the fats into fat acids so as we can get energy. So, and then you can also go into the cell membrane and phosphorylate the nucleus, outside the nucleus, and then go and phosphorylate to call the transcription factors, which can go to the cell membrane and cause what to call the cell division, cause the transcription. That means they can also do what? Phosphorylate. Let me put here. Phosphorylate. One, the precursor factor, the transcription factors, that is TF, and also bring about the protein what? Synthesis, and also bring what called the cell what? Division, and very many others, and also very many others actually. You get. They can also go and phosphorylate the proteins which are important called the muscle contraction. You get the protein, the myosin and actin. They can also be phosphorylated in the muscles, bring about the muscle contraction. What about the DAD here? This DAD here, it is, remember, it is insoluble in water. It is insoluble. What it does, it just remains in cytoplasm. It remains in the cell membrane, and then what it does only is that it is going to combine with the phosphokinase C enzyme. Phosphokinase C enzyme. You get? So this is going to just combine with something called the phosphokinase C enzyme. You get? So this phosphokinase C enzyme here, what is it going to do? It is going to do what we call the phosphorylation. Phosphorylation of what we call now of several or various what? Or various proteins within the cell. So that has been briefly the impact of what we call the pathway of the hormones, most what we call the peptide hormones. Now, this is the same like the steroid hormones. They follow the same thing, except the steroid hormones will talk about something very small next bio lecture. But thank you very much, guys, for listening. This has been our bio lecture. We have seen what a hormone is. We have seen the complex of the hormone. We have seen the receptors where the hormones go. Some hormones bind onto the receptor surface. Some hormones go directly to the hormone, to the inside the cell. Those hormones can bind on the receptors are called the peptide hormones. And these hormones that can go and directly to the cell membrane are called the steroid hormones. The peptide hormones, you've seen that these hormones here, they have receptors, they bind onto them. After binding onto them on the surface of the cell membrane, these receptors here get activated, and then they send signals to the second messengers, which go and amplifying the signal up to the nucleus on the cell or the cytoplasm, and these signals here bring about several cellular processes like the glucose regulation, fat regulation, and very many protein regulation, enzyme regulation, and very many others. Thank you very much for watching. Mr. Goes. Bye bye. We meet.